بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله وبركاته. هذه المحاضرة الأولى من سلسلة محاضرات الانتيبيوتكس دكتور عبد الكريم حميد. إن شاء الله تستفيدون منها بإذنه تعالى. المحاضرة هي عن الانتيبيوتكس. Objective of this lecture is to learn principles of antibiotics and antibacterial drugs, which include classification, effects, clinical uses, and toxicity. This figure shows the bacterial cell with the site of effects of antibiotic. We have the cell wall and the cell membrane, as well as protein synthesis inhibitor, the ribosomes, the effect ribosomes, and some antibiotic affect the DNA or RNA. Others are affect the metabolism process. So, example of inhibitors of cell membrane function are isoniazid and amphotericin B, while the examples of inhibitors of nucleic acid function or synthesis they are fluoroquinolones and rifampin. The inhibitors of protein synthesis are tetracyclines, aminoglycosides, macrolides, as well as clindamycin and chloramphenicol. While the inhibitors of cell wall synthesis, we have the major antibiotic group, which is beta-lactams and vancomycin. The inhibitors of metabolism, we have sulfonamides and trimethoprim. This scheme shows the inhibitors of cell wall synthesis, which include of two major groups, which are beta-lactam antibiotics and other antibiotics. The biggest one is beta-lactam antibiotic, which include penicillins, cephalosporins, carbamazepines, and monobactams. Penicillin include many members. For example, are amoxicillin, ampicillin, gloxacillin, and others. While cephalosporin include 4 generations. The first, according to the introduction in the use in the medical area. First generation, example, cefadroxil, cefazolin, cefalexin, and cephalothane. While second generation includes cefaclor, cefamanidol, and cefurizole, and others. Third generation include cefidinor, cefixime, cefoperazone, cefotaxime, and ceftriaxone. Fourth generation include one antibiotic, which is cefepime. The other group of the beta-lactam antibiotic is carbapinims. Carbapinims include of 3 members right now. The first one is imipenem. Imipenem is not used alone, but should be used with cilastatin, and we will explain why. And we have meropenem and ertapenem. While the fourth group of beta-lactam antibiotics, which is monobactam, it include one antibiotic it is aztronam. Now, other antibiotic, which is inhibitors of cell wall synthesis other than beta-lactam group, we have 2 antibiotic, bacitracin and vancomycin. There are another group in this category, which is beta-lactamase inhibitors. We have glavulanic acid, sulbactam, tozobactam. This group is not antibiotic as their own, but they are help other antibiotic by inhibiting the beta-lactamase enzymes, which is destroyed the beta-lactam group and finish the work of the antibiotic. So it is potentiate and the action of antibiotic. Okay. Beta-lactam antibiotic and other cell wall synthesis We have, this introduction about antibiotics. Some definitions, very important one. Bactericidal, which is an antimicrobial drugs. Bactericidal, an antimicrobial drug that can eradicate an infection in the absence of host defense mechanisms. That means kills bacteria. While bacteriostatic is an antimicrobial drug that inhibits microbial growth but require host defense mechanism to eradicate the infection. It does not kill bacteria. This figure shows us the effect of bactericidal and bacteriostatic drugs on the growth of the bacteria. Beta-lactam antibiotics. Drugs with structure containing a beta-lactam ring include the penicillins and cephalosporins. This ring must be intact for antimicrobial action. If this ring is destroyed, there is no action for no antibiosis action. Beta-lactamases. It is a bacterial enzymes. Examples are penicillinases, cephalosporinases that hydrolyze the lactam ring of certain penicillins and cephalosporin. Minimum inhibitory concentration, lowest concentration of antimicrobial drugs capable of inhibiting growth of an organism in a defined growth medium. While penicillin binding proteins, they are bacterial cytoplasmic membrane proteins, so they are present in bacteria that acts as the initial receptors for penicillins and other beta-lactam antibiotics. Peptidoglycan or murin, it is a chain of polysaccharides and polypeptides that are cross-linked to form the bacterial cell wall. It is very important for integrity of the bacterial cell wall. Selective toxicity, more toxic to invader than to the host, a property of useful antimicrobial drugs. It's very important. Transpeptidase, bacterial enzymes involved in the cross-linking of linear peptidoglycan chains, the final step in the cell wall synthesis. Penicillins and cephalosporins are the major antibiotics that inhibit bacterial cell wall synthesis. Vancomycin and fosfomycin and bacitracin also inhibit cell wall synthesis, but for various reasons are not nearly as important as the beta-lactam drugs because of their side effects and other causes. The selective toxicity of these drugs is mainly due to specific action on the synthesis of a cellular structure that is unique to the microorganism, and that means it is not present in the human cells or in the animal cells. Microbial antibiotic resistance. The emergence of microbial resistance poses a constant challenge to the use of antimicrobial drugs. This is very important dilemma worldwide. Mechanisms underlying microbial resistance to cell wall synthesis. Inhibitors include, cell wall synthesis inhibitors may be resisted by microbes by these mechanisms. The first mechanism is the production of antibiotic inactivating enzymes. So the bacteria will will produce enzymes that will inactivate the antibiotic. And the second mechanism is the change in the structure of the target receptors. Bacteria change the targets of the antibiotics which are present in the bacterial cell to defend themselves against antibiotic. Decrease in the permeability of the microbes cellular membrane to antibiotic. Some antibiotic need to enter the bacterial cell to act against them. So the bacteria will decrease the permeability for these microbes to enter inside the cell. This, table show us the drug resistance types of bacteria to many types of antibiotics. So we have drug resistance due to altered targets, and drug resistance due to decreased accumulation by decreased permeability or increased efflux, and you have drug resistance due to enzymatic inactivation. So examples of the first one, resistance due to altered targets, we have aminoglycosides, chloramphenicol, clindamycin, and others. Examples of decreased permeability, we have fluoroquinolones, beta-lactam, and tetracycline. Examples of increased efflux of antibiotic, fluoroquinolone, macrolides, and tetracycline. While the drug resistance due to enzymatic inactivation, we have aminoglycosides, chloramphenicol, beta-lactam, macrolide, tetracycline. Here it's very important to notice that some antibiotic can be resisted by bacteria by more than one mechanism. For example, aminoglycosides can be resisted by altered targets and enzymatic inactivation, while chloramphenicol can be resisted by altered targets and enzymatic inactivation also. Fluoroquinolone can be resisted by three mechanisms: by altered targets, and by decreased permeability, and by increased efflux. The tetracyclines can be resisted by four types of mechanisms of resistance. By what? By first, altered targets and decreased permeability and increased efflux, as well as the enzymatic inactivation. Strategies designed to combat microbial resistance include what we do, what the researchers, what the doctors do to fight these microbial resistance and make antibiotic act to inhibit or to kill bacteria. The first strategy is the use of adjunctive agents that can protect against antibiotic inactivation. We mentioned, sulbactam and other substances that can, protect antibiotic against the enzymes of the bacteria. The use of antibiotic combinations, for example, the protocols of treatment of TB, tuberculosis, is include 2 to 4 antibiotics in the same times to forbidden the bacteria from developed resistance. The introduction of new and often expensive chemical derivatives of established antibiotics, and this is very expensive. Every antibiotic needs 10 years at least and $100 million at least to be found, for one antibiotic to be found and to be used in success. Efforts to avoid the indiscriminate use of or misuse of antibiotics. This is very important. It's concerned with doctors and pharmacists and the patients also. Antibiotics should be used without need, should be used by prescription under the supervision of the doctors, not by hand buy from the pharmacies.